नमस्ते, हमेशा खुश रहो बिटिया. I'm JV Dr. Louis Reddy, and today I'm going to discuss some diseases of black and green grass. Now, the first disease which we are going to take is Cercospora leaf spot. Now, here you can see it is caused by Cercospora canescens. The symptoms are small circular spots develop on the leaf with gray and center and brown margins. Now, these spots come together or collate together to form brown irregular lesions. Severe defoliation can also occur because it's the brown lesions on the petioles, on the stem, on the leaves, they destroy the photosynthetic area of the leaf and thereby the leaf falls down. The powdery growth of the fungus can also be seen in the center of the spot. Now here we can see. Now, see, this is the severe spot. This is the spot coalescing together. Two spots are coming together. Necrotic area is there. The grayish center shows the fungus and this Cercospora. Now, the pathogen is produced in form of dark brown septate conidia and the conidia are 5 to 6 septate. They are linear in form, hyaline and thin wall. Now, favorable condition is humid weather and dense population. Now, what is the disease cycle? It is spread, survives on the plant debris and clay. And secondary spread is by airborne conidia. For the management of this disease, we have to follow field sanitation, that is, we have to remove and burn the plant debris. We can spray mancozeb at the rate of 2 grams per hectare or carbendazole at the rate of 500 grams per hectare. Now, the second disease which we are going to study is yellow moonbeam mosaic virus. Now, this is caused by moonbeam yellow mosaic virus. Now, the symptom is again, since it is a viral disease and mosaic word is there, the yellow small patches appear on the spots or spots appear on the green color lamina of the leaf. And soon it is developed into golden yellow or bright yellow mosaic. Now, the yellow color increases slowly and the leaves turn completely yellow. And but naturally, if the photosynthetic area of the leaf is being reduced, it leads to few flowers and pods. The pods are small, distorted, and death of the plant can also take place before this leaf settles. So here we see this is the starting phase of yellow bean mosaic virus. You can see the leaves have lost their green color, yellowishness is being seen, and this is the advanced stage where completely yellow leaves are there. The plant is stunted, distorted, and now as said, the pathogen is moonbeam yellow mosaic India virus in northern and central region and moonbeam yellow mosaic virus in western south. Now, the it is a virus belonging to family Gemini virus. Now, what happens here? The Gemini virus particles here double single-stranded DNA, bipartite genome is there with two genomic components, that is A and B gene. Transmission is through whitefly, that is Bemisia tabaci, and it spreads by the feeding of these whiteflies, and summer crop is highly susceptible. Weed hosts also spread the disease as they serve as reservoirs for the knockdown. Management then will be of removing the disease plants and removing of weed hosts, increase in the seed rate. Resistant varieties is the most suitable like EDU10, BBN1 for black gram. And of course, we can follow mixed cropping by growing 2 rows of maize or sorghum for every 15 rows of black gram of maize. Chemical treat control can be done with the seed treatment by imidacloprid 70 Ws at the rate 4 gram per kg or thiamethoxam 70 Ws, and spraying can be done again with these two chemicals, but the rate will be 25 of thiamethoxam will be 25 Wg at the rate 100 gram or imidacloprid at the rate of 17.8% at the rate 100 ml in 500 liters of water. The third disease which we are going to study is anthracnose of beans, that is green gram or black gram, and the causal organism is Coletotrichum lindymucianum. Sexual stage is Glomerella lindymucianum, and the symptoms, they appear on the aerial parts in any state of plant growth. Circular black sunken spots appear with dark center and bright orange margin on leaves and pods. Now, wherever this infection will occur, that part of the leaf will wither. Now, seedlings get blighted due to infection soon after season. Now, here you see this is the initial stage of the infection. This here the lesions are increasing, and this is the form of advanced form of infection. So you see the margins in the color. Now this is a fungus, so it is septate, hyaline, and branch acerulae are there. It arrives beneath the epidermis and rupture through darker septa are also seen. Conidiophores are present. The perfect stage is of perithecia, and 8 ascospores are present, 1 or 2 cells with the central oil group. Favorable condition is more severe in cool and wet season, and the disease cycle is seed-borne. For primary infection, it lives in the infected host tissue in the soil, and secondary spread is by airborne chlamydia produced from infected plants. Now, rain splashes also help in the transfer of the disease. Management is done by hot water treatment at 54 degrees centigrade for 10 minutes. Use of disease-free seeds is there, and crop rotation is followed. Now, field sanitation is the important part of all diseases, that is, we have to remove and destroy the infected plant surface, and chemical control is through carbendazim at the rate of 2 grams per kg seed. Now, the third disease is root rot and leaf blight, or we call it web blight. Rhizoctonia solani and the phylomorph stage is Thanatophorus tutumaris. Now, the disease is caused by seed decay and root rot, damping off, seed blight, stem canker, and leaf blight. Now, this disease is at pod development stage mainly. The fungus causes seed rot, seed blight, and root rot symptoms. So, what happens here? The leaf turns yellow in color, and irregular lesions will appear on the leaf. Now, when they collate such lesions, the big blotches are formed and the affected leaves start to dry prematurely. The root and the basal portion becomes black in color and the bark peels off. So taproot is, when infected, is split open and we see the internal tissues are red or black. Now why is this? Because the xylem and the phloem has been infected by the fungus. Now it is a saprotrophic disease, that means it is occurring in soil, which is primary source, and secondary is through asexual spores, that is humid. And favorable conditions, moist and humid conditions favor the development of this disease. Now, how do we manage this disease? We manage this disease by soil drenching and soil management and through water. So, students, I hope you found my lecture interesting. If you have any queries, please leave the comment section. I will resolve it. This session was powered by Digital Version 2.0